Hello, everybody, and welcome to Chapter 10. In this chapter, we're going to be talking about microbial metabolism, and it's a pretty heavy chapter, so stay with me. If you've taken GOB, which is that General Organic and Biochem course that we offer at OSU-OKC, you've definitely had a little bit of a precursor of what we're going to be talking about in this chapter. We're going to be introducing enzymes and how they work, how they're regulated, as well as aerobic respiration and photosynthesis and those sorts of things in this chapter. So it's going to be kind of a heavy one, and it goes over a lot of information all at once. So try to stay with me. If you get lost in any topics in particular, then please let me know as soon as you can. These are our learning objectives for this chapter. So this will give you some reference into what you need to be focusing on for the chapter. And again, if you get lost on any of these learning objectives or you want more resources to cover a specific topic a little bit more than I cover here, then just reach out and let me know so I can make sure that you guys are getting some good understanding of this content as we are moving through the course. So we're going to start off with the concepts of metabolism. Metabolism is basically, let's zoom out on this a little bit. Metabolism is the whole sum of all chemical reactions and physical workings in a cell, all of them. So making molecules and breaking them down, the whole sum of all of them is metabolism. Making molecules is anabolism. Breaking molecules is catabolism. I remember this because cats tear things apart. Catabolism is the breakdown. Anabolism, like anabolic steroids, is making molecules. If you're making something, you need to put energy in. But when you're breaking something, you can get energy out. So usually we will break things in order to get the energy to build the things. And those things combined are kind of synergistic. But the whole process is metabolism in a cell. A lot of our chemical reactions that go on in our cells aren't able to just happen naturally and spontaneously. They need a little bit of help. Anything that speeds up the rate of a chemical reaction without being consumed in the reaction itself is called a catalyst. And so enzymes are protein catalysts, and most enzymes are proteins, and they are going to help overcome the amount of energy that you need for a reaction to happen at the rate that you want it to and everything like that. So the enzymes help out with that without being used up. Enzymes are organic protein catalysts that help overcome the activation energy of a process. So what do I mean by that? What I mean is whenever we made our DNA, remember how we talked about DNA replication? So DNA polymerase, enzymes often are named with ACE at the end. So DNA polymerase is an enzyme that helps add on complementary nucleotides in DNA. Those nucleotides might just naturally match up A to T and C to G if we just let it happen spontaneously, but we want to have it happen in a controlled and speedy fashion. Then that's what DNA polymerase is there for. It is a catalyst. It is able to help overcome the activation energy to help that happen at the speed and rate and efficiency that it needs to. So that's an example of one of those catalysts. So enzymes, the way that they work, they act on substrates. So the molecule they act on, the molecule they're basically changing or working on is the substrate. Substrates bind at a site on the enzyme called the active site. So enzyme is just a whole blob of protein. The site where the substrate binds into is called the active site, and then the enzyme can act on it. Enzymes, again, are not going to be changed in any way permanently. They can be reused over and over again. And so this makes their role in our cells very useful and less wasteful overall. So the general idea of how enzymes work is that you have in purple here our enzyme, and it acts on this substrate, but not on the other one. It doesn't fit. And this does actually depend quite a lot on shape, not just chemical reactions. Okay, so very heavily on both shape as well as charges, polarities, non-polarities, and all of that. So this is a lot of chemical intermolecular forces, but it's also just general shape. So this can be very predictive as well. So based on the shape of this enzyme, this substrate would fit in, and then we can break it into the two products. The enzyme helps us break it down more effectively and predictably. There are two types of enzymes, simple enzymes and conjugated enzymes. Conjugated enzymes basically are consisting of multiple parts. Simple enzymes are just the protein that does the job. Most of our enzymes are conjugated enzymes, so it's important that we understand their parts. So there's a protein portion. Remember, amino acids come together to create a polypeptide, and then that folds up to create the protein. Conjugated enzymes have that plus something else. Simple enzymes are just the protein folded up. Okay, so what else do we need to make a conjugated enzyme? Conjugated enzymes, the whole functional unit of the protein portion and the thing that helps it is called the holoenzyme. The whole functional enzyme is called the holoenzyme. The protein portion is called the apoenzyme. And the non-protein portion is called the cofactor. Cofactors can be organic molecules or inorganic molecules. Most commonly metal ions. So if it needs copper or if it needs iron or something like that to bind onto it, then that is an inorganic cofactor. If it's a vitamin, like vitamin C or vitamin B12, those are organic molecules. They're not proteins, but they're organic molecules. Remember, what is an organic molecule? It has to have carbon bound to hydrogen, right? So it's an organic molecule that helps the protein do its job. Then those organic molecules that help the proteins are called coenzymes. Coenzymes are organic cofactors. Just cofactors that are organic molecules. So our coenzymes or our cofactors, any of them, our cofactors in general, they bind to another spot on the enzyme that helps that enzyme basically maintain its shape and functionality and helps work on everything. So there's examples down here below where we have, again, the purple is our apoenzyme protein. You might have a metallic cofactor that's hanging out near the active site that helps interact with whatever the substrate is. You might have a coenzyme that just helps stabilize the shape, or you might have both, helping out with different aspects of the protein and how it's going to interact with its substrate. So there's different ways that our cofactors can interact with our apoenzyme to create the holoenzyme for our conjugated enzymes. Okay. Metallic cofactors might include things like iron and copper or magnesium and et cetera. You're hearing a lot about magnesium. Well, your body does need magnesium as a metallic cofactor. So it does require that. It also needs iron and copper and things like this, zinc, whatever. These often help to activate the enzyme and even can interact at the active site to help with interaction of whatever the substrate is. They often participate directly in the chemical reactions. The cofactors may be altered and used up and may not. It just depends on the cofactor. Coenzymes, again, the organic cofactors, often work with the apoenzyme, with the protein, to alter the substrate. So not necessarily directly involved in the chemical reaction, but they help with the action of the protein and how it's going to help the chemical reaction happen. So vitamins are often the best example of what we see the most commonly in our coenzymes. Some organisms secrete exoenzymes. These are literally enzymes that they put outside of the cell in order to break things down around them. Because remember, if we're just talking about bacteria and they're breaking down your body tissues, they're not killing you first and then shoveling parts of you into their mouths. They're too tiny for that. So they have to break down the stuff around them with these exoenzymes, and then they can absorb the particles that are left over from breaking down that stuff. So those are exoenzymes, and they're very commonly toxins to us. So toxins are any molecules that cause damage to us. So many microbes create exoenzymes that are damaging to us and act as toxins. So things like collagenase. So collagenase, ACE being an enzyme, right? It breaks down collagen. And this helps the microbe break down the collagen to use it as a nutrient source, but also helps with the organism being able to get deeper and deeper into your tissues as it's causing an infection in your tissues. Enzymes are proteins, so they are sensitive to denaturation. First, we have to understand kind of what that means. The nature state, natural state, however you want to look at it, normal state of the enzyme that we would call the natural state of it is how it's normally functioning, normally folded and able to do its work. Once we denature it, we are changing the shape of that protein. It often first affects hydrogen bonds. And we have other intermolecular forces like salt bridges and disulfide bonds, which are covalent bonds, that we can then break when we add enough heat or chemicals or high pH and stuff like that. This disrupts the overall shape of the enzyme, and therefore it can't act on the substrate anymore. So denaturation is a very effective tool if you're trying to stop a microbe from functioning. But if you're using heat in the body to denature the proteins, the enzymes of microbes, which is what your fever does, okay? If you're using heat to try to denature them, eventually you can get too hot and denature your own proteins too. Microbes are a little bit more sensitive to that. You have many cells that you can sacrifice in your fight against some microbes. They are just one cell organisms. And so, there's the battle of who can survive the situation the longest that is going on in your own immune system, which we will learn about in Unit 3. But anyways, so yes, so essentially if you can denature or degrade those proteins, they no longer have their shape, then they no longer have their function. They can't bind the substrate at the active site, and then they're no longer, you can control microbes that way and kill them that way. So we'll talk more about how that is used specifically in chapter 11. Okay, that's a Unit 3 thing as well. All right, so moving on. Different types of inhibition in our enzymes. So we have to be able to control the enzyme and how it's working. It can't just willy-nilly, it can't just, your body makes every single protein that it's supposed to, and all of those proteins are working all the time. We need to control when and how they're working. And in fact, some drugs target specifically this side of things. So we have drugs that actually inhibit microbe enzymes, or we have drugs that basically denature microbe enzymes and stuff like that. So how can we control how enzymes work in our body or in the microbes that we might be treating with drugs? One of the ways that we can do that is by occupying the active site. Some inhibitors bind at the active site and therefore prevent the substrate from binding there. Because remember, the active site is the part of the protein where the substrate would bind and the protein would act on it. We can actually block that with inhibitors, and now the substrate can't bind there anymore, and that shuts the enzyme down. But we can also bind at other parts of the protein, which we call regulatory or allosteric sites. So if this is your normal protein, and this is your active site, what's the active site again? The active site is where the substrate binds. Our active site, okay? This is normally where that would bind. If you have an inhibitor that binds here instead, so that our substrate can't bind there, then that is competitive. It's competing with the substrate. So inhibitors that bind at the active site compete with the substrate. All right, so imagine that. With non-competitive inhibition, what happens is an inhibitor binds somewhere else, causes the protein to change shape. So now it's bent in, and now we no longer can bind the substrate. Can't bind there anymore. That's usually what happens. And so you can have inhibitors that bind at the active site. That's called competitive inhibition. Or inhibitors that bind somewhere else, change the protein's shape, and now the active site is no longer available because of the change in the shape. When that happens, that's called non-competitive inhibition because we're not competing for the active site actively. So those are the differences there. Okay. Here are some images that kind of convey that better than my drawing of those two things. Again, on the left, we have the competitive inhibition. Normal thing normally binds there. We act on it. We get our products. When we have something that mimics it, it looks similar. It competes for the active site. It binds there. Now that substrate can't bind there, and we've blocked it. That's competitive inhibition. For non-competitive inhibition, this is our substrate and where it normally binds here to get our product. And then instead of binding at that site, we bound somewhere else, the regulatory or the allosteric site. And that causes a change in the shape of the protein, so we cannot bind. So that's non-competitive. All right, so that's different ways that we can control those. We can also control the expression of enzymes at the genetic level. So that's also another way we can regulate our enzymes. There's a lot of different kinds of enzymes that we have to learn about. These are our 6 major groups of the different groups of enzymes that exist. We do need to kind of be familiar with them. And 4 of these I definitely test on, and we see several of these other ones. The 4 that I test on are these 4. Okay, these other ones are relevant. It's just I don't usually test on them. But oxidoreductases do what they sound like, which is oxidation and reduction. And we're going to talk about that next, so don't worry. But you remember redox reactions, okay? Reduction is gain, right? And then oxidation is loss. There's another way you can remember that. We'll talk about that in a moment, right? But this is referring to transferring electrons. Anytime electrons are moved around, the enzyme that helps with that is called an oxidoreductase. So any enzyme that's involved in moving around electrons, oxidoreductases. We don't always think of electrons as moving around, but if you're moving around a hydrogen atom and it has an electron with it, you moved electrons. Okay, so that's primarily what we are going to talk about them in reference to hydrogens carrying their electrons around. And then whether or not they have the electrons with them or the electrons are free to migrate around with other molecules. That affects those other molecules, which is how the electron transport chain works. And we'll get into that very soon. Okay, transferases are enzymes that transfer groups from one molecule to another. So you can take, a phosphate group off of ATP and put it onto another molecule. Having that negatively charged molecule causes that other molecule to change its shape. And now it does a thing. That's a transferase. Okay. Hydrolases. These are enzymes that cleave bonds on molecules with the addition of water. What it means is we're breaking molecules apart and splitting water while we're doing that. A good example of that might be whenever we take urea. I won't draw the chemical formula for it. I'm going to spare you, okay? But it can be broken down into ammonia and carbon dioxide. That's not carbon dioxide. Dioxide. It's not erasing. There we go. All right. So anyways, ammonia and carbon dioxide. If we have this happening, we often give off water or we break up apart water. I could draw this better. I feel like I could do this better. But anyways, we basically put water in and then we break it apart. We put part of the H's here and part of the oxygens here. The splitting apart of water, lysing the water, hydrolases break apart water in order to break apart other molecules. And so that's basically how hydrolases are going to be used to hydrolyze other molecules. Break water while breaking other big molecules. Okay. The opposite of that are the ligases. So we've already introduced DNA ligase. This is making bonds using water to make bonds. So again, going from nucleotides and nucleotides, when we're linking on new nucleotides onto others, so A, T, C, G, and we keep adding on. So if we had A, T, C, G, and now on the other side, we want to add a T, really, we want to add a T and a A. As we're bringing in the C, and we're linking these guys together on the other side, if we want to join these guys together, that C to the A and whatever to make a big long strand and seal together our Okazaki fragments or seal together any sticky ends or whatever. When we're sealing together those nucleotides, this is a bond called a phosphodiester bond in DNA when this is happening, which is DNA ligase is a ligase that joins those things together. When it does that, it makes water as a product. So it's the opposite of the hydrolases. Hydrolases break water to break bigger molecules. Ligases make water because we're joining together the molecules. It's the reverse process of it. Okay. So there are also lyases, which are involved in double bonds, and isomerases, which have to deal with the orientation of molecules and kind of which orientation they're in. They can change the orientation of the molecule structurally. Okay. So What again is oxidation and reduction? Oxidation is loss of electrons. Reduction is gain of electrons. There's a couple of ways you can remember this. Oil rig or Leo says grr. I learned it as Leo says grr, but you do you, okay? Oxidation is loss. And you have to remember that it's electrons that we're talking about, okay? Reduction is gain. Or lose electrons, E minus is electrons, okay? Lose electrons and you are oxidized. Gain electrons and you are reduced. Gain electrons is reduction. Losing of electrons is oxidation. That's how we're going to remember it, okay? Some of the most important electron carriers that we have that are going to carry electrons for us and then engage with enzymes in our respiration process that we're going to talk about in a minute are NAD and FAD. These guys are coenzymes. What's a coenzyme? A coenzyme is an organic cofactor. What's a cofactor? It is a non-protein molecule that helps a protein perform its enzyme function. Remember the conjugated enzymes. Okay, so that's what that's talking about. So NAD and FAD, what do they stand for? Don't worry about it. I'm never going to ask you. I'm not. You're welcome. We could talk about it. We're not going to. I'm trying to stop myself from getting into it. I don't want to waste your time with it because you don't need to know it. Okay. So, an obvious example of electron exchange, we come back to when we were talking about ionic bonds in chapter 2. We have reducing agents and all of this stuff. But what's really going on here is we have sodium and it has an outer orbital with 1 electron. This is what it needs for its protons and protons to equal out with its electrons. But for its orbitals, having 1 electron in an outer orbital that wants 8, it doesn't need it. It doesn't need it. So it donates it to chlorine. Chlorine has 7 electrons in its outer orbital and would love to have 8. So these guys get along swimmingly. Sodium gives up its electron to chlorine. So with sodium chloride, we now have an ionic bond that has formed. The positive charge, this is not a polarity, this is a proper charge because we've actually lost an electron. If we lost an electron, we are oxidized. Chlorine gained an electron, which is why it's a negative charge here. If it gained an electron, we are reduced because Leo says grr. Okay. So that part is just showing that we've already talked about oxidation and reduction in this class. With our ionic bonds, it happens. Okay. So this is really important because transferring electrons around, we often transfer electrons in terms of a hydrogen. Hydrogen contains a proton, and that allows us to carry an electron around with it. Sometimes we can just be H+. We often see hydrogens hanging out as H+. It don't have an electron. It's just a proton and a neutron. Just 1 proton and a neutron without an electron circling it. 1 proton and a neutron just hanging out is an H+. We call it a proton because the neutron is irrelevant. Okay, so that is a proton and an electron. If we have that, then it's just an H, and it's usually bound to something because it's going to share that electron with somebody else, right? So FYI. Okay, so anytime that we're removing hydrogens from a reaction, it's dehydrogenation. And often we may be doing that as an H+, or we might be bringing the electron with us. And our coenzyme carriers, NAD and FAD, basically can take the H's with the electron or have H pluses come hang out with them. They are allowed to carry the electron around. And so they work as electron carriers. throughout this whole process. That's really all they are, just shuttling around electrons. Why do we need to shuttle around electrons? Because that electron shuttling in the electron transport chain is what's going to power the production of ATP, which is the most important energy source in a cell. Well, what is ATP? It is adenosine triphosphate, okay? So it is an adenine, just like we saw in our ATCGs in DNA and RNA, an adenine, which is a nitrogenous base. It has a ribose. It is not a deoxyribose. So that makes adenosine triphosphate, basically a 3-phosphate version of an RNA nucleotide. Okay, not DNA RNA. Okay, so it's an adenine, a ribose, which is a 5-carbon sugar that we see in RNA, and 3 phosphate groups. It's these 3 phosphate groups that are the key. These are the Big Mac Daddy that bring in the energy. And I'm going to show you how. Basically, those phosphate groups are all negatively charged. They are repelling one another. But especially here, because we got like double repel, because we got both of these guys that are negatively charged. And this last guy does not. Oh, I'm not even showing you this slide. Here we go. Here we go. So you can see that this last guy on the far left out here does not want to hang out with these other 2 negatively charged phosphate groups right next to him. He does not want to come to the party. So when we break those bonds, it releases energy. The most energy comes from releasing this 3rd phosphate, this one here, then the next amount of energy from the 2nd one. So they want to leave. They do not want to be involved in this party. They want to get kicked off, but a covalent bond is forcing them to share electrons with the guy next to them. So they hate it. They hate it. They really hate it, and they want to leave. And when we break that bond, it releases energy that the cell can use to perform work. So ATP, adenosine triphosphate, is the energy currency of the cell, and it gets used up. And so it has to be replaced constantly. And so we can replace it through several different mechanisms. One of them is through substrate level phosphorylation. Substrate level phosphorylation basically makes ATP by transferring a phosphate from something onto something else. This is used limitedly. It does not create a heavy amount of ATP, but it does allow us to create some of it. So transferring a phosphate onto another ATP, and it doesn't want to. It doesn't want to join that party. So it's not always the most favorable choice, but it is one way that we can replenish our ATP. Probably the most commonly and the best way of making ATP is through oxidative phosphorylation. What does oxidative mean? Do you not remember what oxidation and reduction are? Remember that oxidation is loss of an electron. So when we have movement of electrons, oxidative phosphorylation involves adding on an electron, or adding on a phosphate group to ATP, powered by moving electrons. So we'll move electrons through something, and that'll cause us to be able to add a phosphate group. Okay, that's what the electron transport chain does. Another way that we can power adding phosphates to ADP to make ATP is by using the energy from the sun, which is what photosynthesis does. So they usually take like activation. It's the same exact process as the electron transport chain, but instead of being powered by electrons through oxidative phosphorylation, it's powered by sunlight. And then they get that basically activated by photons instead of electrons. So it's pretty neat stuff. So we're going to talk about some of that stuff. All right, so we're going to start off with cellular respiration. And there are different ways that cells can make their ATP through respiration. We have aerobic respiration, anaerobic respiration, and fermentation. Fermentation is a version of anaerobic, but it's not permanent. So if something can ferment to make their ATP, if oxygen is available, they will go through aerobic. It's only without oxygen that they will go through fermentation. Even your own cells can do this. There's a problem with your cells doing it. So if you go for a run or you work out or something like that, and you know how people will tell you, oh, you've got lactic acid buildup. Why did you make a lactic acid? Because your muscle cells fermented, because they didn't have enough ATP available at that moment for that activity that you were doing. So they made more because they weren't getting enough oxygen. They weren't getting enough oxygen, so they made more ATP through fermentation. You cannot sustain your whole body that way, but some parts of your body can sustain temporarily that way. Okay. Overall, we are an aerobic organism. We have some systems that can go through fermentation and often make things like lactic acid as a product, which we're going to talk about. Anaerobic respiration is very similar to aerobic, but it doesn't use oxygen. It uses something else, anything that's not oxygen. Okay, so we're going to talk about that next. All of them share glycolysis. All of them have glycolysis. What is glycolysis? Well, I'm so glad you asked. The three main steps of respiration. So this does come back to breaking it down between aerobic, anaerobic, and fermentation. They all have what? Glycolysis. And this is a very good infographic that basically covers what I need you to know about glycolysis in one view. I need you to know that in glycolysis, we're basically making a little bit of NADH and some ATP. And the product that we're going to be making is pyruvate. We're starting with glucose, and we're making pyruvate too. Pyruvate is then turned into acetyl-CoA in an intermediate step, which is then going to be fed into the Krebs cycle. What's the point of the Krebs cycle? Well, you'll notice really quickly in fermentation, there's no Krebs cycle because in fermentation, all we're doing is glycolysis to make your ATP there. That's it. That's it. Okay. And you can't just keep NADH building up. So the whole point of making lactic acid or ethanol or carbon dioxide from fermentation is to regenerate this NADH back into NAD. So we'll get rid of that H so we're not carrying around electrons unnecessarily. But, you know, we're going to go through all this. We're going to go through all this. Don't worry. Don't worry. Okay. But you can see how they're all very similar. What I want you to notice is that in fermentation, we only do glycolysis. The only difference between aerobic and anaerobic now, between these two, is down at the very bottom. In aerobic, we use oxygen. In anaerobic, we use non-oxygen. Not oxygen, something else. Okay, let's do it. Again, fermentation uses only glycolysis. We're going to come back around to it and break it down a little bit further, but we're going to start off first with aerobic respiration and then talking about glycolysis in general. Okay. In aerobic respiration, it already tells you what is happening. We are using air ultimately. And what does it mean by air? It means oxygen. Our final electron acceptor at the end of the electron transport chain is oxygen in aerobic respiration. So what does that mean in anaerobic? Anaerobic without arrow, no arrow. It means no oxygen as the final electron acceptor. It means something else. Not oxygen, something else. And there's a lot that it could be, and you don't need to know what. You're welcome. Okay. So let's go through it. Glycolysis. The whole point of glycolysis is to convert glucose to pyruvic acid. And it's wild because this is what it looks like. Deep breath. Deep breath. Okay. I am going to simplify this. Follow me. We start with glucose. You need to know that glucose has 6 carbons. It's a 6-carbon sugar. Glucose is a 6-carbon sugar. Okay. Glucose, we start off with 6 carbons in glucose. Okay. We're going to break it apart. And I don't care about any of this. You're welcome. Okay. You do need to know it's a 6-carbon sugar, glucose. And in the end, that what we're making, the whole point of this, the whole point of glycolysis is to make 2 pyruvates. So let me erase this a little bit so we can see our pyruvate. Oh, good. We don't really need any of this other stuff. We don't need glyceraldehyde. We don't need glucose 6-phosphate. We don't need fructose 6-phosphate. We don't need any of that. What we need are these guys. And what are these? Pyruvate. So what did we do? We took a 6-carbon glucose molecule and turned it into 2 3-carbon pyruvates. We ain't adding anything in there, getting fancy, nothing like that. We don't need it. We don't need it. We don't need it. There are steps in the middle of this. It is important to have the general idea that we are making a little bit of NADH. So what is NADH really? This is NAD that is now carrying an electron. So it's an electron source. We made an electron here. Wonder where that's going to go. Okay. The other thing that we did is we did make some ATP. Very little. Net of 2. Make 4, but we use up 2 in the middle of all of this that I've read. Okay. But you get a net of 2. So you get a little bit of ATP. And if you're like, I don't know, is 2 a very little amount? Yes. Compared to what we're going to get in the end, it's nothing. Okay. But if it's all you have, if it's the only way you can make ATP through fermentation, then it's enough. But we're going to make way more in the electron transport chain. But the whole point of this is to get to the electron transport chain. If you're fermenting, you don't have oxygen, you can't do the electron transport chain. You need oxygen to do it. If you can't do the electron transport chain, you don't need to transport electrons. The whole point of the Krebs cycle, which is the next step, is to make electrons for transporting. But if all you need is a little bit of ATP just to stay alive for now through fermentation, glycolysis is enough. So this is glycolysis. The whole point is to make pyruvate. I want to show you something here. It's 6 carbon molecules. And how many do we get at the end? What's 2 times 3? It's 6. Did we lose any carbons along the way? No. Do we make carbon dioxide in glycolysis? No. You have to lose carbons to make carbon dioxide. Don't worry, we're going to do that in a little bit. Okay? So the whole point of glycolysis is to get your 6-carbon glucose sugar and break it into 2 pyruvate molecules. You're like, what? So what's important about pyruvate? It's okay, we haven't gotten there yet. You're going to see. Pyruvate is an intermediate molecule for a lot of things. It can be used in a lot of different pathways. Sometimes all you need is the pyruvate, so you can go through glycolysis just to get pyruvate, which can be used in a lot of different processes. If you're going through fermentation, you can use pyruvate for making amino acids, other sugars, and making fats and things like this. So we can use it for building molecules. It's great. You can go through glycolysis and use the pyruvate to build other molecules for the cell. You're going through respiration, we're making pyruvate so we can make acetyl-CoA to feed it into the Krebs cycle. So we can get electrons to go through the electron transport chain, which is what we're going to talk about. Okay. If you're going through fermentation, then we will take that pyruvate, and because we made an NADH, and we can't just keep NADH sitting around, we need to kick it back into NAD. We need to regenerate NAD. And we'll get rid of that H into either acids like lactic acid, gas. In the process of making those different acids, we might turn that acid into ethanol. We might make carbon dioxide gases, all sorts of stuff. So fermentation, depending on what type of a microbe you are, these might be some products that you would make in fermentation. Facultative anaerobes can just stick with fermentation if they don't have oxygen. If they have oxygen, they're going to go through the whole spiel. Facultative anaerobes. Remember when we learned that? All right. So we said the whole point of glycolysis, the whole point of glycolysis is to go from 6-carbon glucose to 2 3-carbon pyruvates. That's where we left off. Got 2 of them. What now? Then we have this intermediate step where we are turning pyruvate, which is also pyruvic acid, in the body. It gives up its little H plus and it's going to be pyruvate. Okay, the H plus is still bound to it. It's pyruvic acid. Okay. Same thing. So we take pyruvate and we turn it into acetyl coenzyme A, also abbreviated as acetyl CoA. This is the goal of the intermediate step, is to make this. This releases the first CO2 because acetyl CoA, remember we have a 3-carbon pyruvate, acetyl CoA is 2 carbons. So we're losing a carbon. What happens to that carbon that we lost? It comes out as CO2. This is your first reason for exhaling. Your next reason for exhaling comes from the Krebs cycle. Okay. So anyways, the whole point of this is to make acetyl CoA. We also happen to make some NADH. That's it. Okay. All right. We ready? We made a little CO2. We made a little NADH. But the real goal, acetyl CoA. What do we need acetyl CoA for? Well, glycolysis made pyruvate so that the intermediate step could make acetyl CoA, so that the acetyl CoA could be fed into the Krebs cycle. Okay. The whole point of the Krebs cycle is to shred off. I'm trying to get to it. Hold on. Is basically to kick off our electrons and collect them. How do we collect our electrons? By adding them to NAD and FAD. Okay, so we're going to basically take acetyl CoA, which is 2 carbons, and add it to something else. Turns out it's oxaloacetate. Okay, plus something else, which happens 4 carbons, to make a 6-carbon thing that we're going to kick off carbons and electrons in the form of NADH and FADH2. We'll make a little bit of ATP and stuff too, some other stuff. Here's what it looks like. Don't have a heart attack. Okay? Again, the whole point of this, we are starting with pyruvate from glycolysis and turned it into acetyl CoA in that intermediate step. Now that we have our acetyl CoA, which is 2 carbons, we can add to this 4-carbon thing, oxaloacetate. Okay, we add them together and we make a product called citrate. You aren't going to get tested on that, but you've probably heard other professors call it the citric acid cycle. This is why. Okay, Krebs cycle equals citric acid cycle. We're making citric acid. But the whole point of this is that we took a 4-carbon, we added our acetyl CoA, which is 2 carbons, to make a 6-carbon again. Now we're going to shred it. And that's what all of this is. We're going to tear off carbon dioxides and as many electrons as we can. The whole point is to collect electrons. And we're going to put that in the form of NADH and FADH2. Remember, the H's on these guys are carrying electrons with them. So these are coenzyme electron carriers that are going to carry those electrons with those H's. This is the point. We went through glycolysis to get pyruvate so we could turn it into acetyl CoA so we could shred it apart in the Krebs cycle and get electrons. Because you know what happens next. We make NADH here, we make FADH here, we make a little bit of ATP here, we make carbon dioxide here as well. Because we're kicking off carbons in this process. Because again, if we made a 6-carbon thing, and we're going to cycle back through and end up with a 4-carbon thing that we can add another 2 carbons to, and then just keep that cycle going and just keep it going. Remember, in glucose, we get 2 pyruvates. We complete the cycle for each pyruvate. So we do Krebs twice for every glucose that we had. Okay? Okay. Keep up. Because next is the electron transport chain. It's my favorite. Okay? We're going to take all of those electrons from all of that NADH and FADH2 that we made from, from glycolysis, from the intermediate step, and largely from the Krebs cycle. We're going to take all those electrons and turn them into ATP. It's a whole process. If you're moving electrons, though, you got to kind of keep things balanced. And so our checks and balances are why we have to breathe in oxygen. We're going to show in a minute. This whole process is driven by chemiosmosis. This is the idea that our electron carriers shuttle electrons through. They have a negative charge that causes our protons, our H pluses, to cross the membrane. And that this whole process creates the driving force of our oxidative phosphorylation and creating ATP, which is the proton motive force. It is the reason for the season, okay? It is. The proton motive force is basically the difference in where we have our cell membrane here, and it's studded with all of these little guys that they have a name. They're proteins called cytochromes. And at the end of all of this, we have this kind of channel. This channel is called ATP synthase. What do you think it does? Okay. And that what happens is you get NADH and FADH2. I wish I had picked a different color. Let me pick a different color before I get too far with this. I want more of like, let's do orange. That they'll give off H pluses as well as, remember, because they weren't just carrying H pluses, they were also carrying electrons. And the electrons shuttle through the cytochromes. And as the cytochromes get excited, the H pluses get pumped across through chemiosmosis. So we will build up H pluses out here. All these H pluses. I'm going to just put pluses for now, but they're H pluses, okay? Protons. We get an excess of them. This creates a gradient of many H pluses outside of the membrane. So what does diffusion tell you want to do? We just did chapter 9. What does diffusion say? The H pluses want to come back in. They want to be equal on both sides. They want to equal back up. So what do they do to equal back up? Y'all, they come back in. The movement of positively charged H pluses through ATP synthase causes the phosphorylation of ADP into ATP, which is the bring home goal. You're going to make a bunch. You're going to make like 34. Okay? 36 to 34 here, depending if you are a eukaryote or a bacteria. Okay? So you're going to make a ton. Ain't no piddly 2 ATP like we made with glycolysis. You're going to make a ton of ATP here. This is the big goal. And it's all because you pumped H pluses outside of a membrane and created a gradient, which you were only able to do because you brought electrons through the cytochromes, which you were only able to do because you collected the electrons from the Krebs cycle, which you could only do because you had acetyl-CoA from the pyruvate that you got from breaking down glucose in glycolysis. Bing, bang, boom. But you can't let this just sit like this. This is what happens, okay? So now you've got all of these electrons getting shuttled through, right, through our cytochromes. And you've got all these H pluses coming back in. We've got to keep the gradient. We can't let the H pluses come back in and stay there. So we take the H pluses, get a couple, we can't let them build up. We'll take H pluses, pair them with a couple of electrons each. So now they're just H's. Now they can share their electrons. And what are they going to share it with? Please tell me. You already know what they're going to share it with. Half of an O2. What's half of an O2? An O, right? This whole thing is now H2O. You breathe in oxygen only because of this. You breathe in oxygen so you can get rid of your waste protons and your waste electrons. Oxygen is just cleaning up the mess. So oxygen is accepting the electrons here. It's gaining electrons. Oxygen is being reduced here. It is the final electron acceptor in the electron transport chain of aerobic respiration. Okay? Some lovely drawings about this. Here are the pictures of it. Again, it's the same thing that we just looked at. We have our electrons, NADH here, donating the electron that shuttles through our cytochromes, allowing us to pump out our protons, H pluses out here. In bacteria, so we already know in eukaryotes, mitochondria are the powerhouse of the cell. What do you think the powerhouse does? It's this. So in eukaryotes, this happens on the mitochondrial inner membrane, the cristae of the mitochondria. But in bacteria, this just happens on their inner membrane. They don't have mitochondria. Bacteria do not have mitochondria. So where does this happen? It happens on a membrane. It has to. There's only one membrane in bacteria. There's only one. Unless you're gram-negative, there's an outer one. But there's only one usable one for this. It's the inner membrane. The inner membrane is where all this stuff happens. The inner membrane, the cytoplasmic membrane, that's where this happens. The inner membrane. So this is a picture of a gram-positive bacteria. Obviously, it's got this thick cell wall. Okay? So this happens on the inner membrane of the bacteria membrane, the inner membrane. Okay? In eukaryotes, it happens on the folds inside of the mitochondria that we call the cristae. It's still the exact same process. It's just we have an organelle that's dedicated to this. And those folds, we just have a ton of this going on inside of those folds. All cells have mitochondria. Mitochondria is the site of aerobic respiration in eukaryotes. All eukaryotes have mitochondria. All eukaryotes go through aerobic respiration. All eukaryotes do, including plants. Okay? Okay. Anyways, ATP synthase is driven by those hydrogen ions coming through. And because this whole process is driven by moving electrons through the cytochromes to pump the hydrogens out through chemiosmosis, this is generating ATP through oxidative phosphorylation, moving the electrons to drive generating ATP. Again, this is an overview of that whole process that we just kind of walked through. If you want a good big picture of everything that's going on, again, going from glucose to pyruvate, make a little ATP, a little NADH on the way. Pyruvate gets turned into acetyl-CoA, kick off a little carbon dioxide and a little NADH. Okay. Then we'll kick into the Krebs cycle. We'll make a ton of NADH and FADH2, little ATP and carbon dioxide. Then take all those electrons that we collected along the way, shuttle them through the electron transport chain, protons outside, bring them back in through ATP synthase and making our ATP using oxygen as our final electron acceptor. In anaerobic respiration, we just use something else, not oxygen, but otherwise it's the same. That last step is what determines that it is aerobic respiration. If we are using oxygen in that last step, which is right here, aerobic. If we're talking about anaerobic, true anaerobic, not fermentation, in anaerobic fermentation, we might use nitrate or nitrite, or we might use sulfates, all sorts of stuff. Commonly, nitrate can be used here. And we see this in E. coli, for example. Okay. In fermentation, again, we'll just stay stuck in glycolysis to make a small amount of ATP just to stay alive. We'll still make pyruvate, but basically we can't just stop there. After we go from glucose to pyruvate here, we need to get rid of NADH. We have to turn it back into NAD+. We have to regenerate NAD+. And the way that we do that, we kick off that H to either make lactic acid, or we'll kick it off to make ethyl alcohol. We might have some carbon dioxide get shuttled off in there. We might stop at acetaldehyde or whatever. So there's a lot of different ways we can deal with the H, but it changes. We're kicking it back to pyruvate. Pyruvate is what gets changed into ethyl alcohol or lactic acid while we're dealing with the H on NADH. So think of it as this way. Glucose is turned into pyruvate. We also turn it into NADH. To get rid of that H, we feed it back into pyruvate, which then turns pyruvate into lactic acid, ethanol, carbon dioxide, or whatever else that we get from fermentation. So fermentation products. Okay. The idea that you can, in some ways, oops, this is a different slide than what I thought it was going to be. Yes, that you can basically go from glucose to pyruvate. And you can use this to build things, or you can break it apart to make ATP. And then use that ATP to help with the building. This is the idea of amphibolism. Amphibolism is basically pairing together breaking and building and using different intermediates in all of these steps to basically drive each other's reactions. It's that meta idea, they're all working together. And then when you actually take the sum of everything that happened in the cell, that's metabolism. So they are different, okay? Anabolism is building, catabolism is breaking apart. Breaking apart in order to build and working together to do all those things together is amphibolism. And then everything that happened to get there is metabolism. All of this has to happen to build new cells. So anytime you are dividing a cell, building new cells, you need new ribosomes, you need new enzymes, you need energy to build all of that. You need energy to make a new cell wall. You need the components of the cell wall. You need energy to build those components and bring them all together to make, you need a lot of energy. And you need all of the components that are needed to build all the parts. And so amphibolism is basically making the energy to drive the anabolism and building the new parts when you're making new cells. Okay. Let's take a step back to see how our photosynthesis can basically create energy for us. So our phototrophs here, it's very, very similar to what we just talked about, but there's two major steps: light dependent and light independent reactions. Okay. We're going to start with the light dependent reactions. Very, very similar to what we just talked about. We are basically taking a membrane with some, not cytochromes, but in this case, it could be something like chlorophyll, pigments typically. And then this is attached. You remember this, ATP synthase. Instead of shuttling electrons, y'all, light hits it and causes these to get excited, which then allows us to take H pluses out. And we build up H pluses, 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 pluses. It's chemiosmosis, but we're not using oxidation. We're using light to do it. It's not driven by electrons, it's driven by light, but it's the same exact process. The H pluses come through, and now we can make ATP. Otherwise, it's very, very similar, okay? It does not make a ton of ATP. It makes some. It makes enough to power the next step, which are our light independent reactions. The light independent reactions, what happens is you see this ATP, we need to use it. Because what we're going to do with our ATP is we're going to take carbon dioxide through some cycles to make glucose. And you don't need to know the steps. But what you do need to know is we're going to take ATP plus CO2 and some intermediates. You don't need to know what they are. And we're going to turn it into glucose. What's the plant going to do with the glucose? What does it have? Not just chloroplasts. to get this done. Chloroplast, the whole point of the chloroplast is to make the glucose. So we can feed it into the mitochondria for aerobic respiration. Just like that. You brought it full circle. Okay. The plants still need oxygen. They're still going to go through aerobic respiration. They will make some ATP through photosynthesis, enough to make glucose, but they make so much more energy from aerobic respiration in their mitochondria. So that's where most of the energy is going to come from. They still have mitochondria. They still go through aerobic respiration. So they're making glucose here. Glucose can be used for aerobic respiration in their mitochondria, but it can also be used to help build parts of the cell, things like their cell walls and their cell parts, like their new mitochondria or their endoplasmic reticulum, and et cetera, et cetera, et cetera. So they can use it as an intermediate too. So they'll feed it into aerobic respiration, or they'll use it to help build cell parts. But again, they're taking inorganic carbon dioxide and making it into an organic molecule, glucose. That's all fueled by the light from the sun, and they can get more energy to build even more complex things out of that. So it's pretty crazy, and it's pretty unique, and it's pretty impressive too. And all you do is just eat those guys and use that wonderful mechanism that they made organic molecules from and usurped it for your own benefit. So anyways, thanks for joining me in chapter 10. That is it. Let me know if you have any questions, and happy studying.